Hello.
Hello. Yes, sir.
Uh,
We'll start.
Good morning, madam.
Good morning, sir.
So today we are going to discuss about sterilization. Okay.
Yes, sir.
Yeah, please continue.
Okay. Hello, everyone. A warm welcome to today's class at Tawhid Foundation for Allied Healthcare Sciences, Bangalore. In today's session, we'll be focusing on a very important concept in microbiology and healthcare practices, that is sterilization. So, sterilization is a crucial process that ensures complete destruction or removal of all forms of microorganisms, including spores. It is widely used in hospitals, laboratories, pharmaceutical industries, and even in food technology to maintain safety and prevent infections. Before we go into details, remember that sterilization is not just about killing microbes, it's about creating a safe environment for patients, healthcare workers, and research. So, let's begin our discussion with an overview of what sterilization is, why it is necessary, and the different methods used. Let's begin with the introduction. Microorganisms plays an important role in causing infection and contamination. Therefore, sterilization is an important technique in microbiology which helps to remove or destroy microorganisms from materials or surfaces. Common definitions here are sterilization, disinfection, antiseptics, asepsis. Sterilization, it is the process by which an article, surface, or medium is made free of all microorganisms, either in vegetative or spore form. Disinfection, it refers to the destruction of all pathogens or organisms which can cause infection, but not necessarily spores. All organisms may not be killed, but the number is reduced to a level that no longer harmful to death. Antiseptics, these are chemical disinfectants that are safe to apply on living tissues and used to prevent infection by arresting the growth of the microorganisms. Asepsis, this refers to the technique which helps to prevent the occurrence of infection into an uninfected tissue. This refers to the technique which helps to prevent the occurrence of infection. Sterilization method. The diagram categorizes sterilization methods into two major types, physical and chemical. Both play significant roles in eliminating or reducing microorganisms to ensure the safety and sterility of medical instruments, lab equipment, and various environments. We'll start with physical method. Physical method utilize tangible actions or processes rather than chemicals to achieve sterilization. These are further divided into heat, radiation, and filtration. In heat, it is further divided into dry heat and moist heat. For dry heat, incineration, that is burning substances to completely destroy all microorganisms and biological agents. Dry oven, applying dry heat in a dry oven, effective for sterilizing glassware and metal instruments. This both dry heat and dry oven, both methods leads to sterilization. Moist heat, moist heat further divided into pressurized steam and boiling water, water heating, and pasteurization. Pressurized steam, utilizing high pressure steam like an autoclaves to sterile equipment, which further results into again sterilization. Boiling water, water heating, pasteurization, these three lower temperature or non-pressurized moist heat processes. This lead to disinfection, which is not absolute sterilization as some spores may survive in this. Radiation is further divided into ionizing radiation and non-ionizing radiation. Ionizing radiation includes X-rays, gamma rays, cathode rays. These three rays effective for sterilizing heat-sensitive items such as plastics and pharmaceuticals, which lead to result in sterilization. Non-ionizing radiation, mainly ultraviolet light, used to surface sterilization and air purification. Then the third is filtration. Filtration physically separates microorganisms from fluids to airs. Filtration is further divided into three: asbestosis, membrane, and sintered glass. These three used for sterilizing liquids or air that cannot be heated. Starting with chemical sterilization, chemical methods rely on chemical agents to kill or inhibit microorganisms. These are further divided based on the state of the chemical, which is gases and liquid. So, gas, there is two things: formaldehyde and ethylene oxide. Formaldehyde, a gaseous agent used to disinfect and sterilize medical equipment. Ethylene oxide, widely used for sterilizing disposable medical supplies, plastics, and electronics. Liquids, in liquid, four thing is there: alcohol, halogens, phenols, and aldehydes. Alcohols, common disinfectant for skin and surfaces. Halogens includes iodine and chlorine, used for disinfecting water, surfaces, and skin. Phenols, it is utilized in hospital environments for disinfecting surfaces and again equipment. Aldehydes, chemicals like glutaraldehyde, used for high level disinfection of medical instruments. Methods of sterilization. There are two methods of sterilization. First one is physical method, second one is chemical method. In physical method, there is five things: sunlight, that is UV rays, natural germicidal effect. Second is heat, most reliable method. Third is filtration. Fourth is radiation, and fifth is ozone. We'll start with sunlight. Sunlight has an active germicidal effect due to the presence of ultraviolet rays. It is a natural procedure of sterilization which reduces the number of microorganisms in water tanks, lakes, etc. Heat sterilization. Heat is mostly used method of sterilization. Heat is very reliable method. Moreover, it is highly effective and reliable process. There are two major methods of using heat in sterilization, which are dry heat and moist heat. The principle behind both of this method is similar. Dry heat induces the denaturation of protein, oxidative damage, and toxic effect due to the high level of electrolytes. Moreover, the dry heat can also damage the DNA of the microorganism. As a result, the microorganism got killed. Moist heat kills the microorganism by denaturation and coagulation of protein. There are several factors that can influence the heat killing procedure, such as factors affecting heat sterilization. Temperature and duration, inversely related. Microorganism type, spores are heat resistant. Type of material, organic matter, may protect microbes. So we'll start with temperature and duration. The duration and temperature are inversely connected to each other. Therefore, in the case of the long duration of heat provided for sterilization, the temperature will be reduced, while in case of high temperature, the duration will be reduced. Microorganism type, microorganism can be present in both vegetative and spore form. Spore form are generally heat resistant, therefore, the sterilization process will vary on the basis of the characteristic of microorganisms. Third is type of material. Organic substances often provide protection to the vegetative and spore form of microorganism, which reduces the lethal property of heat. Apart from that, the materials containing the substances are also needed to be heat stable for proper sterilization. Dry heat methods. There is four method for dry heat. Red heat, for example, inoculation loops, needles, flaming, glass slides, culture tube mouths, incineration, dressings, carcasses, bedding, hot air oven, glassware, surgical instrument, and powders. We'll start with red heat. In red heat, inoculation loops, wires, forceps, tapes, needles are needed to be sterilized to inhibit microbial contamination. These instruments are held in the flame of a Bunsen burner until they become red hot. Fleming, in this, glass lights, scalpels, and mouth of culture tubes or conical flask are passed through Bunsen frame without allowing them to become red hot. Incineration, this procedure is used to reduce the infective material into ashes by burning. The incinerator is used for the process. Soil dressing, animal carcasses, bedding, and pathological materials are dealt with this method. Hot air oven, so hot air oven, it is widely used method of sterilization by dry heat. The heat inside the oven is maintained by electricity and a fan fitted inside. It provides the adequate distribution of hot air inside the chamber. A thermostat is also connected which maintains the temperature inside the chamber. 160 degrees Celsius for 2 hours is required for sterilization. There are also some alternative temperatures and holding time which include 170 degrees Celsius for 1 hour and 180 degrees Celsius for 30 minutes. Uses of sterilization, for example, glasswares like glass syringe, petri dishes, flasks, pipettes, and test tubes. Surgical instruments like scalpels, scissors, forceps, etc. Chemicals such as liquid, paraffin, fats, sulfonamides, powders, etc. A hot air oven is a laboratory instrument widely used for dry heat sterilization. The diagram provides a labeled view of its main components and the specific functions which are described in below. All these things I'll explain one by one. We'll start with thermometer. I'll tell the purpose as well as its function. Thermometer monitors and displays the internal temperature of the oven chamber to ensure accurate heating. Its placement is usually fixed at the top inside the chamber for easy weaving and precise control. Double wall insulated chamber. Its purpose is maintains heat efficiency by reducing heat loss. The double wall construction ensures the inner temperature remains stable while the exterior stays cooler. Its function is the insulation minimizes energy consumption and protects users from heat exposure. Tray for sample. Its purpose is provide a flat, stable surface to place glasswares. Here you can keep petri plates, conical flasks, etc., metal instruments or other items to be sterilized. Its function is multiple trays maximize capacity and air circulation, ensuring uniform heating. Asbestos door gasket and this timer. For asbestos door gasket, its purpose is to form an airtight seal between the oven chamber and the door. It prevents heat from escaping and helps maintain consistent internal temperature. Timer, it allows user to set and monitor the duration for sterilization. Its function is to ensure that the oven operates only for the required period, aiding in energy efficiency and providing overheating. Temperature and control knob. It is used for setting the desired sterilization temperature and to adjust heating accordingly to requirements, typically from 50 degrees Celsius to 250 degrees Celsius, depending on the load and type of material. Main on and off switch. It turns on the powers of the oven. Its function is simple control to start or stop the oven's operation instantly. High and low heating switch. It lets users select either higher or lower heating intensity. It is useful for different sterilization demands ranging from gentle drying to rapid sterilization. Moist heat methods. Temperature below 100 degrees Celsius. In this, four thing is there: pasteurization, inspisation, vaccine bath, low temperature steam formaldehyde, which is LSLTSF. We'll start with pasteurization. There are two different types of pasteurization methods that are used for sterilization of milk. Holder method, which is 63 degrees Celsius for 30 minutes, and flesh method, 72 degrees Celsius for 20 seconds, followed by cooling quickly to 13 degrees Celsius. This method is effective against all non-sporing pathogens such as mycobacteria, salmonella, etc. Inspisation media like Lonstein Jensen and Loeffler serum are required to sterile at 80 to 85 degrees Celsius for 30 minutes daily on 3 consecutive days. This process is known as inspisation and the instrument used is called inspissator. Vaccine bath. It is used for sterilization of bacterial vaccine at 60 degrees Celsius for 1 hour. In this, serum or other body fluids can be sterilized by heating in a water bath at 56 degrees Celsius for several successive days. Low temperature steam formaldehyde. This method is applicable for materials that cannot be withstand 100 degrees Celsius temperature. In this method, steam at subatmospheric pressure at 75 degrees Celsius with formaldehyde vapor is used. Bacillus styrothermophilus plays an important role as a biological control to test the efficacy of this test. Moist heat at a temperature of 100 degrees Celsius. Here, three things is there: boiling, tenderization, steam sterilizer, which is Coates and Arnold's method. In boiling, it is an effective method that can kill vegetative cells. Boiling for 10 to 30 minutes can kill most of the vegetative cells. However, many spores can withstand this temperature. Boiling can be employed with educate methods and are not available to sterilize glass syringe, rubber stopper, etc. Tenderization. In this case, steam at 100 degrees Celsius for successive 3 days is used. It is also known as intermediate sterilization. In this case, the first exposure kills the vegetative form, and in the intervals between the heating and remaining spores germinates into vegetative form, which are killed on subsequent heating. This process is applied for sterilization of egg, serum, or sugar containing media, which can be damaged due to exposure in high temperature for a longer period. That is why it is taking 3 days, successive consecutive days. Steam sterilizer. Coates and Arnold steam sterilization is usually used for media which can easily decompose due to the high temperature in the autoclave. Those media are kept on a perforated tray and steam at 100 degrees Celsius at an atmospheric pressure passes through the media for 90 minutes. It is an effective method to kill vegetative cells. This diagram represents Coates steam sterilizer. Coates steam sterilizer was one of the earliest devices for sterilizing laboratory and medical instruments using moist heat steam. This diagram shows the main components and their functions, representing a significant milestone in the history of microbiology and aseptic techniques. We'll start with thermometer. Thermometer measures the internal temperature of the sterilizer. Its function is to ensure that the water inside reaches the required temperature, typically above 100 degrees Celsius, for effective sterilization. The thermometer is usually inserted through the lid at the top of this container. Lid Lid seals the top of the sterilizer chamber. Its function is to keep steam from escaping and maintaining pressure and temperature. The lid is typically tightly secured and may include blow-off valves for security. These valves, two valves. Water gauze. Water gauze shows the water level inside the device. It allows user to monitor and adjust water content ensuring enough is present to generate steam throughout the sterilization process but preventing overflow. Ring of flames. It provides heat for boiling the water inside the chamber. It usually connected to a gas source. This ring heats the base evenly to generate steam, which rises upwards and fills the chamber. Moist heat, temperature above 100 degrees Celsius. For moist heat, we'll start with autoclave. Steam above 100 degrees Celsius or saturated steam has a better killing capacity than dry heat. Bacterial proteins coagulate rapidly at moist heat. Saturated steam has the ability to penetrate any porous material. When steam comes out into contact with the cooler surface, it condenses into water and releases its latent heat to the surface. The large reduction in volume sucks in more steam to the same side and the process continues until the temperature of the substance raised to that of steam. The condensed water produces moist conditions for killing the microbes present. So the autoclave is a modified pressure cooker which contains a vertical or horizontal cylinder. The cylinder is made up of stainless steel. A lid on the cylinder is placed and fastened by screw clamps to make it airtight. The lid contains a steam discharge unit, a pressure gauge, and a safety valve. Moreover, a thermostat is present to monitor the temperature. Heat is produced by electricity. At the time of sterilization, the cylinder is filled with an adequate amount of water and is kept for some time for preheating. After that, the material which are needed to be sterilized are inserted into the cylinder and the lid is then closed tightly. The temperature will increase eventually along with the pressure. When the temperature reaches 121 degrees Celsius and the pressure is at 15 psi, the sterilization is performed for 15 minutes. Uses of sterilization, you can use culture media, rubber material, dressing gloves, materials that are unable to withstand dry heat in a hot air oven. Ozone sterilization. Ozone sterilizer uses oxygen, water, and electricity to produce ozone within the sterilizer and provides sterilization without producing toxic chemicals. It runs at it runs at 25 to 35 degrees Celsius temperature. Inside this device, the oxygen is converted into atomic oxygen due to the intense electrical field. The atomic oxygen is then combined with the oxygen molecules to produce ozone. The ozone provides a sterility assurance of 10 to the power of 6 in approximately 4 hours. Filtration. This process is useful for sterilizing those materials which are unable to withstand heat. There are several types of filtration such as candle filters, asbestos disc filters, sintered glass filters, membrane filters, air filters that is HEPA, syringe filters. For, for candle filters, it is used for purification of water. These filters consist of hollow candles and water passes through the candles for purification. Asbestos disc filters. These are made up of magnesium silicate. Sintered glass filters. These are prepared by fusing finely powdered glass powders. Membrane filters. It is approximately 0.22 μm. These are made up of cellulose esters and are used for water analysis, sterility testing, and for the preparation of the solution. Membrane filters are available in pore size, for example, approximately 0.015 to 12 micron. The 0.22 micron here filter is most commonly used as it is smaller than bacteria. Air filters. These filters are used in laminar airflow chambers to give bacteria free air supply. These are also known as high efficiency particulate air, which is HEPA filters. These filters can separate particles of 0 micron or larger than that. Syringe filters, syringe fitted with the membrane of different diameters are available. A limitation of using the filtration process is that the pores are not small enough for viruses. Radiation. Radiation is further divided into two: radiation and non-ionizing radiation. Ionizing radiation include gamma rays, X-rays which damage DNA used for syringe catheters. Non-ionizing region, ultraviolet light damages DNA used for OT and laminar airflow. We'll start with ionizing radiation. Ionizing radiation such as gamma rays, X-rays and cosmic rays are used for sterilization process. Due to the high penetrating power, these radiations are lethal for cells. The bacterial cells are killed by damage in the DNA, which is mentioned here, DNA damage. The bacterial cell are killed by DNA damage. Gamma radiation from a cobalt-60 source are commonly used for sterilization of disposable items. This procedure is also known as cold sterilization. Non-ionizing radiation, infrared radiation and UV radiation comes under this of radiation. Infrared radiation is used for mass sterilization of syringe and catheters. UV radiation with a wavelength of 240 to 280 nanometer has bactericidal capacity. The UV radiation causes protein denaturation and interferes with DNA replication of bacteria. UV radiations are used for sterilization of closed areas, surfaces, operation theaters, OT, laminar airflow, etc. Chemical methods key properties. Chemical method, severe chemical agents are used as antiseptic and disinfectants. The properties of a chemical antiseptic or disinfectants are following. The chemical disinfectants need to have a broad spectrum of activity against of all microorganisms such as bacteria, viruses, protozoa and fungi. The chemical agent should act in the presence of organic matter. High penetration power is an important property of the chemical agents. The chemical agents need to be chemically stable under both acid and basic conditions. The chemical substances should not have any corrosion activity in metals. The disinfectants are needed to be non-toxic if absorbed into circulation. And last, the chemical agents are needed to be easily available and less expensive, cheap and easily available. Chemical agents, for example, alcohols, ethyl alcohol and isopropenyl alcohol. These two alcohol are frequently used as chemical agents for disinfection. Both of the chemicals facilitate the protein denaturation of bacterial proteins. 70% ethyl alcohol is the standard concentration which is used for disinfection. These are used as skin antiseptics. Apart from this, methyl alcohol has actively against fungal spores and used to disinfection of inoculation cabinets. Aldehydes, in aldehydes, formaldehyde, glutaraldehyde, opium. We'll start with formaldehyde. It is known for its bactericidal, sporocidal, and virucidal activities. It can be used in both aqueous as well as gaseous form. A 10% formalin solution is a standard chemical disinfectant. It is used for prevention of tissues for histological examination, sterilization of bacterial vaccine, preparation of toxoids from toxins. Glutaraldehyde, it has its activity against bacteria, Mycobacterium tuberculosis, which causes TB, fungi, and viruses including HIV, hepatitis B, etc. It can also kill spores and is known for its less toxic nature, it is used as a 2% buffered solution. Glutaraldehyde, it is used for sterilization of cystoscopes, endoscopes, and bronchoscopes. Sterilization of plastic, endotracheal tubes, face masks, metal instruments, etc. OPA is nothing but orthophthaledehyde. It is stable high level of disinfectant and it is known for its stability during storage. It has bactericidal effects against mycobacteria. 0.5% OPA is slowly sporicidal and OPA vapors irritate the respiratory tract and eyes. Therefore, it must be handled with appropriate safety. Phenols and its derivatives. Phenols Lister, father of antiseptic surgery, used phenol for the first time in the sterilization of surgical instruments. Phenols work as a disinfectant and kill microorganisms by cell membrane damage. It is toxic for the skin. Different derivatives of phenol are used as antiseptics, which are following: crasols, which is known as Lysol, chlorohexidine, Savlon, chlorooxylenol, Dettol, hexachlorophene. So we'll give one example. Crasols Savlon is an example of chlorhexidine solution which is widely used in wounds pre-operative disinfection of the skin. It is bactericidal at high dilution. Moreover, it has fungicidal activity. Dettol Dettol is commercially available as a chloroxylenol solution. It is less toxic and less irritant. Hexachlorophene, it is bacteriostatic and very high dilution. Halogens. Halogens, chlorine and iodine are commonly used disinfectants. Chlorine is used in water supplies, swimming pools, food, and dairy industries. Chlorine compounds in the form of bleaching powder, sodium hypochlorite, and chloramines. The disinfection action of all the chlorine compounds is due to the release of free chlorine, which become a stronger oxidative agent. Iodine in alcoholic and aqueous solution is used as a skin disinfectant. It is active against Mycobacterium tuberculosis and slightly active against spores. Compounds with iodine with surface active agents known as iodophores are claimed to be more active than aqueous or alcohol solution. Oxidizing agents: hydrogen peroxide, para, parasitic acid. Hydrogen peroxide, it is an effective against most organisms in the concentration of 3 to 6%. However, it kills spores at higher concentration, which is 10 to 25%. The mode of action is by the liberation of free hydroxyl radical on the decomposition of hydrogen peroxide. These free radicals are active ingredients in the disinfection process. Parasitic acid, it is stronger than H2O2. It is an oxidizing agent and is more potent germicidal agent than hydrogen peroxide. Salts and dyes. Salts, slats of heavy metals have a toxic effect on bacteria. The salts of copper, silver, mercury are used as a disinfectant. They are protein coagulant and act by combining with sulfidyl group of bacterial proteins and other essential intracellular compounds. Merthiolate, which is sodium ethyl mercury thiosalicate, is used in a dilution of 1 is to 10,000 for the preservation of serum. Dyes, two group of dyes, aniline and acridine dyes, have been this aniline, which is crystal violet, acridine, acriflavine, have been used as a skin and wound antiseptics. Both the dyes have bacteriostatic activity. Aniline dyes include crystal violet, brilliant green, and malachite green. Acridine dyes include acriflavine, coflavine, proflavine, and aminocrine dyes. Vapor phase disinfectants: ethylene oxide, ETO, beta propiolactone, BPO. Ethylene oxide, it is a colorless liquid with a boiling point of 10.7 degrees Celsius. It is effective against all types of microorganisms, including viruses and spores. It acts by alkylating the amino, carboxyl, hydroxyl, and sulfhydryl groups in protein molecules. In addition, it reacts with DNA and RNA. It is specially used for sterilizing plastics and rubber articles, respirators, heart-lung machines, dental equipment, etc. Beta-propylolactone. This is a condensation product of ketane and formaldehyde. It has rapid action and used in 0.2%. It is more efficient in fumigation than formaldehyde. PPO is used for the inactivation of vaccines. Summary. um, we learned about sterilization and its methods. So I'll give a short overview. There are two main categories of sterilization method: physical methods and chemical method. Physical method, these rely on agents like heat, filtration, radiation, and ozone. Heat, such as dry heat, such as hot air oven; moist heat, such as autoclave, boiling. Filtration, filtration using membrane filters to remove microbes. Radiation, ionizing radiation like X-rays and gamma rays, or non-ionizing like UV, ultraviolet light. Ozone, oxidizing agent with strong sterilizing power, sometimes grouped with physical and chemical methods. Chemicals, alcohols, aldehydes, phenols, halogen, etc. Alcohol, example, ethanol and isopropanol. Aldehydes, formaldehyde, glutaraldehyde. Phenols, halogens like chlorine and iodine compounds. Choice of sterilization method always depends on the type of material, example heat sensitive versus heat resistant, the type of microorganism, some are more resistant like spores, and purpose, the intended purpose, for example surgical instruments, culture media, pharmaceutical, and food preservation. For question and answer, 3 mark questions, 5 questions is there, and for 5 marks 5 question is there. So for 3 mark questions, define sterilization. In this, you can tell what is sterilization, how does sterilization differ from disinfection. Second is differentiate between antiseptic and disinfectant. So, what is the difference between antiseptic and disinfectant? Where are antiseptic and disinfectants are commonly used? You can elaborate. Write 2 uses of hot air oven. What are the 2 common uses of hot air ovens in a laboratory or medical settings? Give principle of moist heat sterilization. What is the principle behind moist heat sterilization? How does moist heat can kill microorganisms? Name biological indicator used in autoclave. Name the biological indicator used in autoclave sterilization. Why is this biological indicator are important? Further, you can tell in detail. For 5 mark, for 5 mark, there is 5 question. Explain methods of sterilization by dry heat. So first, what is dry heat sterilization? You can describe the methods used, for example, hot air oven, flaming, and incineration. Describe, and then you can explain principle behind dry heat sterilization, oxidation of cell components, mention typical temperature and the time settings for sterilization, and lastly you can discuss the advantages and disadvantages of dry heat sterilization. Describe pasteurization with types. Here you can first define what is pasteurization and its purpose. Explain how pasteurization works. For example, mild heat treatment to reduce microbial load, and then discuss different types of pasteurization. For example, low temperature, long time, LT, high temperature, short time, ultra high temperature, you can mention here. Write short note on autoclave with diagram. Here you can define autoclave and its uses and sterilization, explain the working principle, steam under pressure, for example at 121 degrees Celsius for 15 to 30 minutes, describe its key parts of an autoclave, importance of autoclave, and include a neat, well-labeled diagram of an autoclave. Compare ionizing and non-ionizing radiation. You can compare differentiate between ionizing and non-ionizing radiation. You can give characteristic features, its nature, mechanism, penetration ability, its application, and last examples for ionizing and non-ionizing radiation. For example, gamma rays, X-rays, ultraviolet radiation. You can mention it here. Lastly, explain chemical methods of sterilization with examples. Here, define chemical sterilization using chemicals to kill microbes. Describe common chemical statement: ethylene, formaldehyde, glutaraldehyde, alcohols, halogens. Give one examples of each. You can explain mechanism of action, oxidative damage, protein denaturation, alkylation, and lastly, application where it is used for medical instrument, heat sensitive instrument, etc. Thank you.
Thank you so much, madam. It
Thank
was
you,
very
sir.
nice explanation regarding sterilization and its types.
Thank
And
you, sir.
actually, coming to glutaraldehyde,
Yes, sir.
best one for Mycobacterium tuberculosis, as
Yes, sir.
well as fungus and virus, I think.
Yes, yes, sir.
Yeah, and coming to one more, that is halogens, iodine.
Yes, yes, sir.
It is also useful for MTB, Mycobacterium tuberculosis.
Yes, sir.
And one more, hydrogen peroxide. It is best to kill spores.
Force. Correct, sir.
Percentage is, I think, 10 to 25%. Yeah,
Correct, sir.
yeah. And it was very nice presentation, madam. And
Thank
coming
you.
to my question is here.
Yes,
So
sir.
autoclave, the instrument which
Yes,
is
sir.
used in hospital,
Yes, sir.
will you mention the locations where it will be helpful, where it is used?
Location. I'll do that,
Okay.
sir.
Locations in the sense, for example, laboratory. One is laboratory.
Correct,
There also we
correct.
can use.
Yes, sir.
And one more is in CSSD.
Yes, sir.
CSSD is the department where all material, which are
Mm-hmm.
non-sterile, yeah, unsterile
Correct.
material can be gathered, yeah, pulled up.
Mm-hmm.
So
Mm-hmm.
there, those material will get steriled. Okay.
Right.
So the main purpose of this autoclave, yeah, other instruments is to get sterilized properly. So then we can use for further purpose.
Further. Yes,
Yeah,
yes, sir.
further use.
Mm-hmm.
Yeah, thank you, madam. Thank you so much for
Thank you,
your
sir.
clear information and proper, what we can say, class.
Yes.
Okay?
Oh,
And you
thank you,
have
sir.
covered a lot today. Okay,
Thank you
thank
so
you,
much.
madam. Thank you so
Thank
much.
you. Thank you, sir.
So shall we wind up, madam?
Yes, sir.
Yeah, thank you, madam.
Thank you, sir. Thank you.